Welcome all. I'm David Scadden. I'm a Jordan Professor at HMS and the Director of the Center for Regenerative Medicine at Mass General. I'm delighted to be running this panel on the common blood disorders and cell and gene therapies for them. We have a fantastic panel of people who are participating. Leschly Kean, who is the Stranahan Professor at HMS and runs the Cell and Gene Therapy Program at Children's Hospital and Dana-Farber Cancer Institute. Samarth Kulkarni, who is the CEO of CRISPR Therapeutics, leading CRISPR company. Nick Leschly, the CEO, also known as Chief Bluebird of Bluebird Bio, with things in the clinic approved for treatment of these disorders. And Mike McCune, who's been a long-time colleague and friend who has worked on early days of stem cell gene therapy and extensively evaluating new kinds of technologies that might make a difference, particularly in resource limited environments where things like the sickle cell disorders as well as HIV are such common and devastating problems. So let me just start with you, Leschly. I mean, you see patients all the time. We know that there are cell and gene therapies that already exist for people with sickle cell disease. Allogeneic transplant has been a long known cure. Do we need new cell and gene therapies? And what do you look for that will change this field so that you can have a different conversation with those patients and their families over the next 5 years or so?
Yeah, thanks so much, David, and thanks for inviting me to be on the panel. It's great to be having this conversation. So I, as you said, have run the stem cell transplant program at Boston Children's and Dana-Farber for kids. And actually, my whole career has been inspired by sickle cell patients. They are the most common patients that I used to transplant during my training in Atlanta, or think about transplanting. So the short answer to your question is absolutely we need better therapies. The field of stem cell transplant has the ability to cure some of the most at-risk patients, both in terms of malignant disease, but really what we're really focused on more here is non-malignant disease. And for those kids, let's just focus on the non-malignant disease. Transplant has the risk of graft versus host disease, which is not acceptable for those patients. And so what we've done in transplant and the work that I do in my lab is all directed towards trying to prevent and control graft versus host disease, but it looms as the killer, the most common non-relapse cause of mortality for our patients. And so that is the big barrier to transplant. Because the risk of graft versus host disease is directly related to the risk of HLA disparity in transplant donors, this really opens up that unmet need for either autologous products or, the next sort of phase of in vivo gene therapy. And so there's a huge unmet need, and it's incredibly exciting that we're actually at this precipice right now.
Yeah, and I mean, Nick, you've got success with people and you've seen the incredible ability to turn people's lives around. I have to say, like you, Leschly, having taken care of these people, and I'm sure Mike has the same experience, these are people who are burdened with one of the worst medical curses they could ever live with. And so to see that turn around, Nick, must be incredibly gratifying. And can you just give us a sense, you've also seen some of the dark side of what can happen here. So can you give us a sense of what challenges and what opportunities you're seeing?
Sure, and thanks again for also having me here. It's great to hear you, Leschly, and others sort of reaffirm just the absolutely remarkable need that's out there, and also the coming of age of the promise of gene therapy and cell therapy that, David, you've been working on for a long time, right? But now it's hitting reality. And so you're right. We do have a product that's approved for thalassemia, and we have some data in sickle cell and others that really are starting us to really kind of inspire not only the bluebirds that have helped on this, but everyone around it, right? When you can get a 99% reduction in events in pretty much all the patients we treated in sickle cell, that starts to kind of change your frame of reference of what's possible. But you're absolutely right. In a pioneering space like this, there are many more things we don't know and we don't understand. So how do we stay humble in the face of these advances and realize that good is not good enough, right? So as good as the therapies are, I think you're referencing some of the more recent cases. We had 2 cases in our sickle cell groups that had one case on MDS, another case on AML. And the first question is, my goodness, is this one of these existential events for gene therapy in the cell therapy space? And I have to give credit to a lot of the scientists, particularly at Bluebird and a few other places outside, that really just cranked down and realized this is not anything to do with the vector, we don't believe, as you look at this. What it does reflect is a really, and Leschly, you know this better than I do, but a really, really profoundly complicated marrow in sickle cell. And the fact that they have this stress on the marrow that is causing a high replication demand to kind of deliver what the body needs. And that also leads to a higher potential for malignancy. And that's what we think we're seeing here. And we start off these events. You said the dark days, right? I actually think darkness leads to light. We've seen it again and again and again at Bluebird, where we face this existential, oh shit moment. And it actually teaches us a remarkable amount. And even this short period in the last month and a half, I think we've learned more about sickle cell patients, more thought leaders are engaged, the NHLBI, the FDA, everyone is crashing the boards on behalf of this community. So actually, as dark as it is, it also now is showing us, well, actually, you know what? We now starting to understand more and more what's going on and what approaches like myself and Sam and others are working on can do better. Right? Because it's already pretty doggone good, but how do we make it better? So that's another way of looking at, I'd say, the dark times.
Yeah, Nick, I love your perspective on this. And I have to say much credit to you and all the folks at Bluebird of being so open and engaged with the community and the NHLBI, and to try to really figure out what to do to make this so that we don't incur this kind of downside. But so Sam, you also have the amazing success of seeing patients get better through this, but there are issues, of course, in terms of getting people enrolled and getting them through the early parts of the process. From your perspective, what do we have to do to make this become something that's more widely adopted, even in a place where the resources are not the challenge, like in the US?
Yeah, thank you for having me on this panel. It's a pleasure to be among this august company. I have to say that this is one of the most exciting things that we may look back on 20 years hence and say, this was sort of the period where we figured out how to make cell therapies, smart, intelligent therapies that will cure. It's a new paradigm in medicine versus the paradigm of treating symptoms or chronic care. And first of all, I have to, give kudos to Nick and the Bluebird team and others, a lot of researchers in the NIH, at the NIH and other places that did a lot of work that built up to this. Boston Children's had a big role in identifying BCL11A, which is one of the targets we're going after. But the results are just remarkable. I think so far, both in the Bluebird trial, the Boston Children's trial with David Williams, our trial together with Vertex, you're seeing cures. These are patients who are living with pain and go to hospital 7-8 times a year with the chance of death each time. And this is truly transformational. That said, I think the system needs to come together to enable this and bring this to patients in a widespread manner. The enrollment challenges for me, those are typical challenges. They're surmountable. And over time, you get a flywheel effect. You get a momentum as you get sites on board and teach them how to use these therapies, and enlist the top experts. But the key challenge, and I suspect will be the one that we're going to see play out over the next 2-3 years, is the system willing to pay for these therapies, particularly for a patient population in the US and in Europe, which has been ignored for a very long time. And while there's willingness and people always say that they want to support patients with sickle cell disease, it's going to require new mechanisms of payment from CMS and other agencies at the state level. And that'll be the biggest test, I think, for our society. Are we going to rise to the occasion now that science has provided an option to bring these therapies to patients? So that's number one. The second, I think, I'm sure Mike will talk more about it, is the global access, right? I think this is the first step, and we eventually need to get to all the sickle and thalassemia patients across the globe. There's thalassemia patients in India, China, Southeast Asia. There's sickle patients in Africa, India, and other places. And the autologous ex vivo cell therapy will have some limitations in how far it can go because of the cost involved. But that said, I think, you're chipping away one at a time. And that global access is going to be a second piece that can only be solved with improved technologies and perhaps new ways of doing gene editing or gene insertion. But I think the more immediate challenge in the near term for us to learn is this access question, which is an important question both for the US and Europe.
Yeah, and of course, where the disease is most common, of course, is in the place where these kinds of technologies will be least accessible and least affordable. And so, Mike, you've been thinking hard about this. I mean, there are, 300,000 children born every year with sickle cell disease worldwide, most of them in sub-Saharan Africa. And you've focused so much on HIV disease, which is also largely based there, and millions of people affected. So what do we need to do? How do we break the bottleneck?
Right, great questions, David. And I also like to thank you and the organizers for putting this all together. As you pointed out, I'm an AIDS doc, and I have been, like you, actually since the early 80s, taking care of patients with HIV and trying to make them better faster and for longer periods of time. And we thought initially that highly active or combination antiretroviral therapy would do that, but it's just not the case. There's millions upon millions of people, many of them in low and middle income countries like sub-Saharan Africa, that cannot access or cannot stay on antiretroviral therapy. So over the past couple of years working at the Gates Foundation, we've asked the question, how can we actually help those individuals in a way that would allow them to have durable suppression of HIV absent having to take antiretroviral therapy? When you think about it, if you imagine the lack of infrastructure there, the intervention would likely have to be provided as a single shot, done and gone, as Jay Bradner said earlier today. You'd have to target long-lived cells. Who are they? Hematopoietic stem cells, T-stem central memory cells, B cells in the body. They have to be done in vivo, and it would have to introduce a modification that leads to a quote-unquote cure for HIV disease. So put simply, what we're talking about here is in vivo gene therapy focused on self-renewing cells of the hematopoietic system. When we started this work, we're thinking about a 10 to 15 year time frame with a stepwise series of scientific steps that really are going through the following bodies of work. First, we have to figure out what the design of a cure might be. And frankly, we don't have a clue for HIV at this point. There's some glimmers of ideas, but there's not much. So in order to think about how to actually tackle the second problem, that is targeting and editing hematopoietic stem cells, we decided that we would work on sickle or other diseases of hematopoietic stem cells first as a pathway to figure out what to do for HIV. And so be it by viral or non-viral vectors, we're now asking the question, which ones work best? And then secondarily of those, which ones are actually developable? Which ones can be brought into resource-limited parts of the world? Thirdly, we know that we have to start talking to people that are there, listening to them, engaging them in this work, getting them ready for it, and listening to their ideas and incorporating that into what we were doing. And not least, we knew from the get-go that we would have to do this in partnership with others. The Gates Foundation could not do it alone. We have to bring in other expertise, the appetite for high risk, and not least importantly, other resources. So over the past couple of years, since the program was initiated, we have made a number of investments in a number of academic labs and small to mid-cap biotech companies, including some of those on this call right now, to motivate research towards in vivo gene therapy exclusively targeting hematopoietic stem cells. In 2019, we entered into a 50-50 collaboration with the NIH, NIAID, NHLBI, Office of the Director, exclusively focused on in vivo gene therapies for sickle and HIV going to resource-limited parts of the world. And then late last year, we announced a substantial collaboration with the Novartis Institutes of Biomedical Research, standing up a research team within the hallways of NIBR and Cambridge, focused again exclusively on developing, testing, and bringing in vivo gene therapies for sickle, and then if that is to work, HIV into these countries. The science is actually moving a lot more quickly than I would have imagined in the beginning, and there seems to be a fair amount of enthusiasm and appetite to take the risk. And I think part of that is because even though it's true that most people with HIV and sickle live in resource limited parts of the world, as you point out, there are many in the States, so there are commercial markets here. And finally, we believe that the lessons learned from this will be applicable to other disease indications, say, for instance, the introduction of CAR T cells for treatment of hematologic malignancies and solid tumors.
Yeah, yeah. So, one of the, it's remarkable that there's been this confluence of attention on the problems now, and the a way in which multiple different related technologies are being used to deploy gene modification strategies. But I have to say that it's a little concerning that the targets seem to be really very concentrated. So, Sam, how do you think about this? Is there too much attention on one particular set of targets? Do we need to be thinking more broadly than is currently the approach?
Now, when you say targets, David, do you mean indications we're going after, or targets within
Indications? I think, within the indications, certainly there are a couple of other strategies for sickle cell. There are multiple. Your approach is very different than Nick's, but I just wonder whether or not the blinders are still a little bit narrow.
No, absolutely. I think, I think the reason there's so much such gravitational pull towards sickle is it's a disease that's been ignored for a long time. There's no other therapies. These patients have no other, really no other options. And I think it's one of the most well-known genetic diseases with reasonable prevalence. But I think that's the key is I think if the system can figure out sickle cell, you're going to see a number of other indications that are going to come through behind it because, you can imagine that you could insert genes into CD34 cells for hemophilia, for instance. You could put any factors in, especially if you can do this in vivo, then, you've taken it to a whole different level of convenience and ease without having any of the safety issues. And you could imagine both common and rare diseases with CD34 cells as a biofactory. So I think there'll be a lot more behind it, but that risk is hard to take when you don't know if the system can absorb sickle cell. I think it's even greater risk to do everything else. I think there has to be that incentive system built in with the reasonable number of patients and payer systems and countries willing to pay for it that would advance the science in this case. And by the way, it's a great model system to learn the technology and improve upon it. For instance, we're developing gentler conditioning agents for sickle cell and thalassemia that could actually pave the way for HIV.
Right.
Right now with the medicines available out there for HIV, CD34 transplant is not a practical solution. But if you have a gentler conditioning agent on the back of sickle cell and thalassemia, all of a sudden you've enabled a dramatically different therapy or cure for HIV patients. Yeah.
Yeah. Well, and I have to say, I love your perspective that by what we learn through sickle, we'll be able to actually apply to a wider range. And I have to say, I think, given that we know that blood cells go everywhere and that they are a very smart system for being able to deliver different compounds, they may actually become much more of the living medicine that we hear about with CAR Ts than we currently exploit. I mean, Leschly, you deal with kids with lots of different genetic disorders. I mean, the cell therapies have been a transformation for many of them. What's the horizon? What should we be thinking about next generation?
So I agree with everything that's been said so far. And I think most importantly, if we think about gene editing and other genetic modifications that we can make to stem cells, developing it as sort of a cassette approach, right? That, as you say, once you sort of figure things out with the potentially most common or most underserved sickle cell kids, there are so many smaller, more rare diseases that could be cured. And I'll get to some of them in a second, but I think it's very important to think about what are the risks that underlie all of these. And I think, Sam, you just said one of the most important things, which is that if you think about what are the barriers to the prevailing treatment that we now give our kids, which is allogeneic transplant. I mentioned GVHD. The other big issue is genotoxic conditioning. And that is, if we can solve genotoxic conditioning, first of all, it makes that nearer term autologous stem cell transplant approach for gene therapy way more tenable. I believe that we need to drive towards in vivo gene therapy, but boy, if we're going to make, and I think the experience of the sickle cell patients on the sickle cell gene therapy trials have now shown this, that it is an absolute critical unmet need to identify non-genotoxic conditioning agents. And that is because, what we've found in the last several months is that what we really didn't very much appreciate before is that there will be some diseases like sickle cell that have a revved up marrow that will be at more risk potentially of marrow inflammation and marrow disease, including leukemia, if we give them genotoxic conditioning. If we can solve that, then essentially any disease that either is caused by a blood cell or can be delivered, a cure can be delivered by a blood cell, will be amenable. So this includes, ALD, it includes all of the inborn errors in metabolism. If you think about the developing world, I do a lot of work in Cambodia, and hemoglobin E is the most prevalent hemoglobinopathy in Southeast Asia. There is a whole host, and when you think about the untapped potential worldwide of children who grow up with chronic anemia, it's clear that we could make a leapfrog in terms of our ability to sort of support health all over the world. So really the world is our oyster, but we've got to get past this genotoxic conditioning if we're going to do it.
Well, Leschly, you're singing my tune as well. I mean, I just really think that's a critical issue. But I mean, Nick, you've learned that it's not just the conditioning. What you found was not a conditioning related problem. It's the underlying biology. So are you thinking that we are getting lessons that will apply, or we're going to have to do this disease by disease?
No, I mean, it's fascinating to hear, because I think this is kind of like an and a conversation, right? Which is, I think everybody's right and everyone's probably equally wrong in that sense, right? Because we just don't know. But I try to break it down a little bit differently, as thinking horizons. And I think Leschly and Sam both said this, which is, let's get to as many patients as we can, learn as much as we can with what we have in front of us that's tangible. There's an incredible amount of lessons on how to deploy this thing, how to manufacture this thing. And I mean, there are lessons. I call it the Bluebird, and I think probably the field advent calendar, right? Every morning you're opening something up, right? And you're like, oh shit, what happened today, right? So there's an element of that, but there's also an element of this. That doesn't mean you don't think about the next horizon and the third horizon, which I'd put probably in vivo on the third horizon, and arguably the most important, right? So we got to make advances towards it because it's most global. But we are learning that it's not as simple as that. One of these cases might have had a busulfan conditioning related. The other one definitely didn't. Why? Because the cells were sitting in the freezer when the patient got treated with it. So there are other things going on here and how they come together. So we think of it as sort of in the context of the tools and the rules, right? We got to understand how the tools are starting to establish rules, and then how we can partner them with a layered approach, because none of us, right? There's no gene addition approach that's the answer. There's no CAR T or TCR. There's no genome editing approach that's the answer. Our view on that is it's a layered approach, right? These are going to be molecular combos that we put in, whether it's ex vivo or in vivo, it doesn't really matter, but you're not going to get an answer in here that takes into account all those. So that is just an ability to ask and answer and learn as fast as we can. And I think we're just getting to the point where we have the capabilities to do that. And so in there, we can get deeper into the aspects of what's actually we believe going on, but it comes down to what Leschly said, I think in particular as relates to sickle, is just that replicative stress leads to a situation. Yeah. And it can be exacerbated maybe in ways we don't even appreciate yet. But what we also know is the average life expectancy of sickle cell is mid-40s in the US. That's not okay, right? So it is not exactly an area where you shouldn't be willing to take some risk here, because that's not. So we just have to work through it. And I think everyone's trying to partner. And that's part of the reason we're trying to be as open as we can, because if you're open, you actually get openness back. And we're learning a boatload from the way people are looking at our data, as long as people are reasonable about it. So our big concern was, would the FDA freak out? They didn't freak out. They were totally controlled, and they were great partners in this. And so was the NHLBI.
No, that's great. Great. So Mike, we're dealing not just with the issues of technology and problems of understanding how best to deliver this, but also how to deal with it in the context of different cultural settings. And what do you think about this? How do we deal with all the multiple regulatory agencies and cultural mores? And how does this actually become something that can really have the impact that penicillin did?
Yeah. I mean, it's hugely important, as we learned from the rollout of the COVID-2 vaccines, and before that, the rollout of PrEP, right, for HIV, which we've had for a decade, but still isn't taken up. And I like the idea, I like Nick's idea of thinking about successive frontiers in the future that we can strive to reach, although I'd also like to see your advent calendar someday. But I think right now we know we're not going to have in vivo gene therapies for HIV and sickle anywhere for some time to come. But we know now that we need to engage the communities and the places to which these therapies will go sooner than later, like yesterday. And who are those stakeholders? It's not just the ones you named, right? It's the care providers, it's the regulators, it's the ministers of health, it's the pharmaceutical companies, it's the payers, it's the faith communities. And then, not least, it's the patients and their families. And we've been actively engaged in doing that over the past couple of years. And as a consequence of a convening that we held in early 2019, the foundation has helped to set up a public-private partnership called HCAP, HIV Cure African Acceleration Program, which you can read about in an article that was published in The Lancet earlier this year, which is led by Mark Dybul of Georgetown and Zuzu Sikazwa of Zambia, meant to bring together these key stakeholders to engage that discussion. Okay, that has to happen. Right now, too, we've published just recently the first target product profile for relatively complicated cures for HIV, but this public-private partnership is now developing TPPs for ex vivo and in vivo gene therapies that could be taken anywhere, including to low and middle income countries. But at the same time, there is a very committed and energetic coalition of physician scientists and patient advocates led by Sissy Kitio as a joint clinical research center in Kampala and Jen Adair at the Hutch, banded together to form an entity called the Global Gene Therapy Initiative, working not just in sub-Saharan Africa, but also in India and elsewhere, where a number of hematopoietic diseases are present, hoping, they say, to bring ex vivo gene therapy onto the field within 2 years. It's fantastic. So all of this is moving quickly. We know that it needs to happen. And I'll end by saying, it's not just the low and middle income countries we need to think about, right? In the United States as well, we need to educate people.
Our neighbors, you bet.
It's just, this discussion has to start. And the sooner the
great. And I'm afraid I'm about to get the electronic taser. We've used our time, but I want to thank you all. You've been fantastic. You guys are really leading the way and changing the world. Bravo. So I thank you. I'm sure that the community in need thanks you even more. It's great.